Hello everyone, welcome to lecture 8 where we're going to be discussing genetics, regional variation, and biogeography. In lecture today, I'm going to challenge you to think about what you know about a species. What is a species? What is a subspecies? And then today I really want you to focus on understanding the 3 major factors that influence the geographic distribution of both amphibians and reptiles. So when we think about biogeography and what causes amphibians and reptiles to be distributed where they are, we know that climate, resources, and interspecific variations vary from area to area, and that each population can adapt to its local condition. And remember, we talked last time about how sexual breeding populations tend to have better adaptive potential than our asexual breeding populations. So we know each population will adapt differently and eventually diverge genetically or evolve from other populations. If this divergence is allowed to continue, speciation will occur. Speciation, however, is a really rare outcome because adjacent populations oftentimes exchange individuals, allowing for things like gene flow to occur. So therefore, the rate of gene flow is a function of 2 things: the closeness of the populations and the dispersal tendency of the species. And we're going to talk about each of these in detail. But first, let's talk about species. So today, in today's definition, species are distinguished by differences in things like body functions, biochemistry, behavior, and their genetic makeup. And if you think about our biological definition, our classical biological species concept definition, it states that you have genetically distinctive populations of individuals that are isolated reproductively from all other populations. So genetically distinct, reproductively isolated. Those are the 2 key factors in our biological species concept, and that's important to remember. There are numerous alternative species concepts, and the last time I checked, there were 27 to 30 different species concepts. For purposes of this class, we're going to still rely on that biological species concept, again, which relies to genetically distinct and reproductively isolated. And here you see at least 3 different alternative species concepts that, again, I'm not going to go over in great detail, but note that there are lots and lots of different ways in which you can define a species. But we will adhere to the biological species concept, the definition for which I've already given you. Well, how do subspecies come into play? So there's lots of different definitions of a subspecies as well. And subspecies designations are actually really common in herpetology. And they're also a source of quite a bit of disagreement. So one definition of subspecies is a taxonomic subdivision of a species. Definition 2, a population of a particular region that is genetically distinguishable from other populations, yet it's still capable of interbreeding with them. An alternative subspecies definition, a grouping of organisms that differ from other members by color or size or other morphological features. A classic example of a subspecies here in Indiana would be our race runners in the family Teiidae. We have our prairie race runner on the left and our 6-line race runner on the right. So we have Aspidosceles sexlineatus viridis for a prairie race runner, which tends to have males with blue hues on their venter, whereas our Aspidosceles sexlineatus, our nominate subspecies, tends to not have that blue coloration on the venter in males. Well, now I'd like to introduce you to a new concept called a cline. A cline is a gradual and continual change in a character, and the character can be almost anything: size, some sort of color morphology difference, but it's a gradual and continual change in some character by a series of populations throughout the range of that species. It's usually along a geographic or environmental gradient. And oftentimes, when you look at a cline, the individuals or populations on the 2 extremes of that environmental gradient or geographic gradient tend to differ considerably. So let's take a look at a schematic that might help explain this concept a little more. This colored bar represents a group of natural populations. These different colors represent clinal variation in a species or a group of species. Now, this cline of populations could stretch across a continent, or they could bend to encircle a lake. An example for a coast-to-coast cline would be our tiger salamanders, which occurs from California all the way to the east coast of the United States. Now, these small yellow areas on this schematic are areas of hybridization. So these are separating the species or subspecies along some hypothetical cline by this hybridization. Now, this hybridization could be a couple of kilometers, or it could be very extensive and encompass several hundred kilometers. Again, it depends on the natural history, the biology of the species that we're talking about. So let's take a look at real life examples. So here we have the plastral markings of our painted turtle. So we have Western forms that have a very intricate pattern on their plastron. You can see that these areas of pigmentation extend well onto the edges of the plastron, whereas our Eastern painted turtle or Midland painted turtle have this area of pigmentation restricted along the midline of the plastron. So this is our Midland painted turtle. This is our Western along the edge of the plastron, restricted to the midline. There's an area of hybridization between these two subspecies where you get areas of the plastron that are somewhere marked between a Western and a Midland. So remember, the Midland is restricted to the midline, and the Western, this dark pigmentation extends well and beyond the edge of the plastron. And in this hybrid, you see it comes close, but not quite. So here we have our Midland, which is this area of yellow, which includes most of the typical Midwestern states. And then the Western painted turtle occurs in the upper plains area. This area of brown is the hybridization zone between the Midland painted turtle and the Western painted turtle. And it just so happens that if we find a vaccination for COVID, and those of you that are wildlife students and you're able to go to camp, this area of hybridization occurs right where we do our turtle trapping for summer camp. And so you have a really great opportunity to see examples of this clinal variation. Where you, in this hybrid zone, you'll see some individuals that tend to resemble more Western turtles, Western painted turtles, and other individuals that tend to resemble more of their Midland painted turtles. So it's a really cool hybrid zone, and it's a really great example to show what this might look like in nature. Well, let me introduce another question to you. So I want you to think for just a second, how many of you like that biological species concept that we defined early on in this class, in this lecture, which is, again, your populations are genetically distinct and they're reproductively isolated. So think about that definition for a minute and say, yep, that's a definition that I could stand behind. That makes sense to me. It's very clear cut. So take just a minute and think about whether or not you think the biological species concept is clear cut. Now, how do you define a species when the individuals don't interbreed, but they all stem from one population? And this is known as a ring species. So this is a really interesting example of how complicated the species concept can quickly become. So let me show you on this schematic what I'm talking about. So imagine populations of the species A. So here's species A, and all these circles represent populations. These subpopulations, A1 through A5, A sub A through A sub E, all stem from this original population, population A. Now, population A1 and A2 have this area where they can interbreed. A2 can also interbreed with A3. A3 can also interbreed with A2 and A4. A4 can interbreed with A3 and A5, and so on. And the same can be true for the pattern on the bottom. A sub A can breed with the nominate population A as well as AB, and then AB can interbreed with populations that are its nearest neighbor. But when you get to the very end of this ring species, A5 and AE. Remember, all of these stem from A. A5 and AE can no longer interbreed. Even though they come into contact with one another, they no longer interbreed. So the question is, are A5 and AE, which are just subpopulations of A, are these now different species? Because they're no longer able to interbreed. There's no longer any genetic exchange between A5 and AE. So with regard to the biological species concept, are A5 and AE different species? So I want you to think about that for a minute. Are there any management implications that might result from a phenomenon that exists like this? So think about that for just a minute. And then I want you to put yourself into a real life scenario. We actually have this very phenomenon occurring in nature. And it happens to be the distribution of Ensatina eschscholtzi. These are some plethodonid salamanders that occur along the Pacific coast. And this particular group of species occur from British Columbia in Canada all the way down to Baja of Mexico. There are 7 subspecies that are currently recognized and all occur in California. There's considerable variation among the color patterns of all these various subspecies. So here you can see the schematic of all the different subspecies and how dramatically they vary in terms of their physical appearance. They have different, very obvious, different color patterns. But what's most interesting about this species of salamanders is that the 2 southernmost subspecies meet in several locations, but they don't hybridize. So here we have the Great Basin, or the Great Central Valley in California, and this cluster of 7 subspecies completely encircles the Great Valley of California, again forming a ring. And you have these areas of nearest neighbor that hybridize with one another, but in this zone where you have these 2 subspecies being locally adjacent to one another, they rarely, if ever, hybridize with one another. So the question again is, are they separate species? Does this warrant elevating those 2 subpopulations, those 2 subspecies, to species status, when again, they can also breed with their nearest neighbor? So again, think about what that means from a management perspective. Think what that means from a protection. If one of these subspecies becomes endangered, does it warrant a protection under the Endangered Species Act? Is it its own species? Do you protect the subspecies? All these are real life questions that wildlife managers have to think about. So again, I want you to be thinking about this in terms of our biological species concept. All right, let's talk about some morphological variation. Let's talk about latitudinal changes within a species, specific to changes in body weight. If you think about Bergmann's rule, it states that animals have a tendency to be larger in polar regions, medium size in temperate climates, and smallest in our tropical ones. So does that sound familiar to you? In a previous lecture where we talked about a concept known as the surface area to volume ratio. So why might this play out, right? So we know in some amphibians and reptiles, this pattern holds true. There are some exceptions, but those species that have exceptions also have cryoprotectives, right? And we talked about that as well. So in colder place, an animal can get larger without overheating. Or alternatively, you can think about it is that in a colder place, an animal must get larger in order to avoid becoming too cold. And all of this has to do with the surface area to volume ratio and the animal size as it relates to Bergmann's rule. You also have changes in body color. So think about those animals in really hot environments with a lot of sunshine, what types of colorations they may have, right? Because we know dark color tends to absorb light, whereas a light colored animal reflects that light. So, and the light in this case could equal heat, since we're talking about ectotherms or poikilotherms. In the last couple slides, I want to talk about biogeography. Biogeography refers to patterns of geographical distribution. So we've talked a couple of times about some of the factors that limit the distribution of amphibians and reptiles. So think about the natural history of all of our various groups that we've talked about. There are three major factors that influence the geographic distribution of herps. The first is climate. Animals will not occupy areas where their physiological tolerances will not be exceeded. So remember, think back to our lecture on ATR, that activity temperature range. We know we have thermal maximums, we know we have thermal minimums, and we know that those maximum minimums exist from the day the eggs are deposited all the way up until the adults are senescent. So animals will not occupy areas where the physiological tolerances will be exceeded. And in amphibians, there are three key factors: temperature, rainfall, and their periodicity. So how frequently does it get warm or cold? How frequently does it get hot or dry or wet or dry? So their temperature, rainfall, and the periodicity as it relates to climate is a major factor that influences the geographic distribution of amphibians and reptiles. The second, the availability and access to resources. Animals need to be able to maintain their energy requirements, that is, find food, and reproduce, to have access to adequate nesting substrates. And then lastly are the dispersal abilities. Small fossorial amphibians and reptiles tend to have poor dispersal abilities, whereas large aquatic animals tend to be very good dispersers. And we have an entire lecture coming up right after this one that focuses on dispersal and some characteristics that are important to dispersal. And I want to leave you today with these last two slides. These are species density contour maps for the continental United States. And here I want you to pay attention to where you see amphibian species hotspots. So you'll see areas where there's very few amphibian species, might be in this intermountain zone, maybe it's in the southwest. So think about what geology occurs in those particular areas. What is the climate like in some of these areas versus areas where there tends to be a lot of amphibian diversity? What tends to be the geological features that you see here? What tends to be the climate and the dispersal abilities of species that might live there? So I want you to compare the amphibian species diversity contour map, what I'm showing you right now. Think about what is driving this particular pattern, and then compare it to the reptile species contour map. This looks very different. You'll see areas that are occupied by reptiles, and in some cases, heavily occupied by reptiles where there was almost no amphibian diversity. And then you'll see areas where there's both amphibian and reptile diversity. Again, think about the geology, think about the climate, think about the types of species that occur in those areas, and be able to have a conversation and a discussion of what might be driving this biogeography of our herp species. So that's it for lecture 8, and join us next time for lecture 9, where we're going to be talking about movements, home range, territory, and habitat selection.